Hi, everyone, and welcome to episode 792 of the People Behind the Science podcast. Today, we're excited to be resharing our interview with Dr. Barb Katzenback. Listeners, Barb studies how frogs defend themselves from diseases in order to stay alive. And she also investigates how the environments that frogs live in can impact their ability to defend against diseases. We know that populations of frogs and other amphibians have been in decline, but they are critical components of the food web. They're important for pest control, and they're indicators for the health of their environment. And they also produce some interesting molecules on the surface of their skin that may have relevance for human health. And in this episode, Barb talks more about her research and her life outside the lab. So I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of the People Behind the Science podcast. Today, I am delighted to introduce you all to our guest researcher, Dr. Barbara Katzenback. So, Barb, thanks so much for joining us today. How are you?
I'm fine, Marie. Thank you so much for having me.
Absolutely. Well, we're excited to learn more about you and the wonderful work you do. But let me first take a moment to introduce you in a little bit more detail to our listeners. So listeners, Barb is an assistant professor in the Department of Biology at the University of Waterloo. She earned her bachelor's degree with honors in Immunology and Infection from the University of Alberta. She then received her PhD in Physiology, Cell, and Developmental Biology, also from the University of Alberta. And Barb was awarded a Natural Sciences and Engineering Research Council, or NSERC Postdoctoral Fellowship, to conduct research at Carleton University, followed by an NSERC Banting Postdoctoral Fellowship to conduct research at the University of Waterloo before she joined the faculty there. And in our interview today, Barb is going to tell us more about her life and her science. So Barb, we'd love to hear about life first. Can you tell us, what do you like to do when you're not very busy doing science?
I try to do a number of things, Marie. Probably the first is physical activity. So I like to go out running, do yoga, hiking. Those have been kind of some constant activities in my life, but I also like to try different things. So I've played soccer, I've done spin classes, cross-country skiing, rock climbing, even had a little bout of inner tube water polo.
Ooh.
And of course, on my jaunts or my hikes, I like to take my husband and my 2 dogs with me, and we have a lot of fun. And I find having creative outlets are also really important. So I do some pottery. Sometimes I do a little bit of art, crafting, baking, gardening, sewing, that kind of thing. And of course, I try to get in some reading for pleasure when I get the chance, but those moments are a little bit few and far between at the moment.
I completely understand. Well, it sounds like you have got a lot going on. And I must say, I am fascinated by this inner tube water polo. I've never tried it myself. What's it like?
It is so much fun, but hilariously awkward. So it's essentially a giant inner tube that you would maybe use for sledding down a hill, but it's in a pool. And you're essentially playing water polo, but you're sitting in an inner tube, and you have to use your arms and legs to paddle you around this giant pool and then try to throw the soccer ball or volleyball into the net. So I'm a relatively short individual. I'm only 5'4", and these inner tubes are quite large. So you can imagine only a very little bit of my arms and legs are peeking out from this inner tube, trying to frantically paddle in the water. But it was great fun. We did it with a group of friends with beer afterwards. So that was always a highlight.
This sounds awesome. I must say, I have to try it sometime. And you mentioned reading as well as a hobby, and this is something I'm always trying to fit more into my own life. So I love getting book recommendations from everybody that I talk to. So Barb, I'm going to ask you, do you have a favorite book, whether it's a science book or a non-science book, that you've just really enjoyed and think me and our listeners should read?
Well, I am from the generation of Anne of Green Gables fans. Mm-hmm. So that would probably be my all-time favorite. But I recently reread The Stone Angel by Margaret Lawrence, and I got even more out of it the second time than the first time. So I recommend all of those people who haven't read it to definitely give it a read, because I think it's consistently ranked one of the best Canadian books ever written.
Awesome. Well, we will add Anne of Green Gables series and also Stone Angel to our reading list for our listeners out there. And Barb, we've talked a little bit about your life outside of science, but I would love to chat about your research next because I know you're doing some really cool stuff in the lab. So can you tell us how you describe your work to someone who's maybe not familiar with your particular field or your research?
Or I guess I would say that I normally start off with something that maybe they would know about. So I talk about worldwide declines in amphibian populations that has slowly been appearing more and more in the news. And we talk about how important amphibians are to our daily lives. So people normally equate frogs with great for pest control, eating those mosquitoes or grasshoppers. And then they also can identify them as being important in the food web. So we start off understanding how frogs are important to our everyday lives. And then I tell them, well, one of the big underlying reasons behind amphibian declines is their susceptibility to diseases that are emerging in their environment. And I study how frogs defend themselves against these diseases so that they don't die. And I also look at how the environment that the frog lives in impacts their ability to defend against these diseases. And I often get the question from people that I'm talking about, why is this important? So we touch on ideas in the conversation about how frogs are really sensitive animals and they act as sentinels or indicators of the health of the environment or ecosystem that they're living in. So often I equate this to the canary in the coal mine. Often if amphibians are having problems, that means really there's something wrong on a larger scale. We also touch on the idea about how we can transfer a lot of what we learn about the frog immune system to that of the immune system of humans. And I also talk about how frogs can produce a lot of really awesome molecules on the surface of their skin. One of them is known as antimicrobial peptides that they use as weapons to combat disease. And there's been some interest in maybe developing some of these antimicrobial peptides as use in human medicine, perhaps as agents to fight against some of our own diseases.
Very cool. Well, Barb, you're doing some awesome work. And I think you brought up a really important point here that many of us are hearing about on the news, these amphibian declines. But can you maybe give us a big picture perspective of what is the extent of these declines that you're seeing?
Well, it's global, so it's all over the world. It's not like it's our neighbor's problem. It's happening here in Canada as well, and the US. And about 1/3, or some estimates are even higher than that, of amphibians are declining worldwide. So that's a pretty substantial proportion of amphibian populations that are endangered or threatened or considered extremely vulnerable to becoming extinct.
Certainly. So definitely a really important issue to be investigating. And I look forward to chatting more about some of these projects that you're working on as we go through our interview today. But I'd love to talk a little bit first about what motivates you. I think there's so much to do in a day. And we mentioned all the things that you love to do, even outside of science as well. It can be hard to balance everything and stay motivated. So Barb, do you have a favorite quote or a saying or even a force that really resonates with you and keeps you motivated?
Well, I think in science, you're faced with a couple of things, or at least I am. One is constant failure. You're trying to figure something out that you know nothing about, so you have to try many different things. A favorite quote from Albert Einstein is, if we knew what we were doing, it wouldn't be called research.
I love that quote.
Yeah. That always reminds me it's okay to not know what you're doing. You're just trying your best to figure it out. And then I'm sure there's a longstanding version of this other phrase that was coined by someone else, but I first heard it from my PhD advisor, Mike Belosevic. And he said, if you don't play, you can't get. So what he was really saying is don't discount yourself before others weigh in and say that you're not good enough. And that was really eye-opening for me in grad school, during my postdocs, and even now as an assistant professor, because sometimes scientists, we all suffer from imposter syndrome, and sometimes we discount ourselves from opportunities before we even apply for them. So anytime I feel like I'm getting into that zone, my advisor's words ring in my mind, and I'm motivated to keep trying and keep putting myself out there.
I love it. Such an important message. I'm glad you brought it up. And you mentioned this impactful quote from your graduate advisor. And I think these advisors and mentors can be such a great influence on our careers as scientists. But are there other role models or mentors or people that you want to mention today who also had a big impact on you?
I've been very fortunate to have so many wonderful mentors along the way. And it's hard to say that any one person, because it really is a collection. I've learned from all of them, things that I like about them and things that I wasn't maybe as fond of, but I really got to learn about myself through them. I would say it's super important to have representation. I think one of the things that first sparked my interest in science was I was reading I think it was either a junior high or high school chemistry textbook, and they had a spotlight on Maud Benton, who was a Canadian physician and biochemist. And she was one of the scientists responsible for studying enzyme kinetics that led to the Michaelis-Menten equation. She hadn't done just this one scientific contribution, but she had done so many. And I was just in awe of the powerhouse that this Canadian female scientist was. And I just remember thinking, I really want to be like her. And this was early on. But I would say probably the most influential role model would be my PhD advisor, Mike Milosevic. He's a professor at the University of Alberta, and he studied and still studies host-parasite interactions. And at the time I was an undergrad student, he was regarded as one of the best professors by the students in the Honors Immunology and Infection program. And you look at him, you're, wow, he's just this amazing scientist and so full of enthusiasm, zest for life. And he really put his students first. So I was motivated to go in and pursue grad school with him. And I still talk to him regularly, and he continues to mentor me even till today.
Well, that is awesome, Barb. It sounds like you've had some great mentors and influences over the course of your career. And I love that you highlighted this featured Maud Benton in your textbook, who really sparked your interest in science. I think these early experiences and exposures to science can have such a big impact on our careers. Was this the first time you maybe started getting interested in science? Or are there other memories as well that really capture that moment where that spark was lit for you?
I always enjoyed biology and chemistry, and I always liked to tinker with things as a kid. And you don't think about it at the time, but I think all people are naturally curious. And it's just sometimes we get more cultivation than others. So it might be a little bit more of a product of our environment. But as a child, I remember that I would do experiments with beetles.
Oh, really?
Yeah. So in our house, there were these window wells. And in the window wells, they were filled with rock. And underneath the rock, there would always be these black beetles. And I thought to myself, it's only rock and dirt here. How are these beetles possibly surviving? So I would capture the beetles and then put them in ice cream buckets with different types of components from the window well. So one would only have nothing. One would have soil. One would have rock. One would have soil and rock. And then I would leave them out and see how long the beetles would live to try to understand what it was that they needed in their environment to be successful. That was one of the things that I did. And I think it was so great in junior high and high school. We would have labs associated with our chemistry and biology and physics courses. So you really got to get in there and tinker with things. And I just loved that. I loved tinkering.
That is awesome. I love this idea of doing some experiments in your own home as a kid. And I think that is a great way to explore and learn about your world. So now, can you tell us, Barb, what then took you from this curious kid starting to do laboratory work in high school, maybe junior high as well, to where you are today? How did you find that path to your current position?
So I think a lot of it is just being open to experiences and not worrying too much about the future, but just enjoying where you are now and finding what you like to do now. So because I knew I liked high school labs, I actually stumbled upon a early research experience. It was as a kid in Edmonton, Alberta. There was a Heritage Youth Researcher Summer Studentship Program, or the HIRES program. And as a grade 11 student, you could apply for it. And they would select, I think, about 20 high school kids or grade 11 kids. And they got to go into a research lab at the University of Alberta for 6 weeks and work with a professor doing science. So I was extremely lucky. I got selected as one of the lucky students to go and do this. And I actually got placed in a nursing lab with Dr. Carol Estabrooks at the University of Alberta and just started doing some dry lab science. So more dealing with data and how to analyze it. And I really enjoyed the environment and, again, asking the questions and getting some sort of answer at the end. So then I kind of pursued that. After grade 12, I applied for another summer studentship, but this time it was for 4 months through the Alberta Heritage Foundation for Medical Research. And I, again, was exceedingly lucky and I got one. And this time I was working in a lab in a kind of oncology pharmacy with Dr. Mercer, studying radioactive drug targeting to tumors. And that was a wet lab science. So it was manipulating mice, injecting them with different drugs, seeing where those drugs went. Hopefully they were targeting it to different locations in the body where the tumor was located. And I just really enjoyed the scientific process. So when I was in undergrad, I specifically tried to seek out research opportunities. I originally started out actually in the honors biochemistry program, then switched afterwards to the honors immunology and infection program. So I just kind of kept looking for research opportunities along the way. So I also worked in a chemistry lab, and then I did an undergraduate honors thesis project before starting a master's degree with Mike Belosevic, which I then switched up into a PhD. And then it just kind of went from there.
Gotcha. And then we mentioned you had these wonderful opportunities, the NSERC Postdoctoral Fellowships. Can you tell us a little bit about those?
So I actually decided to go to Carleton University because I heard Dr. Ken Story talk at a conference. I was there giving a talk myself. It was at the Canadian Society for Zoologists. Ken Story is a biochemist, and one of the animal models that he studies is the North American wood frog, which, as you may know, is this amazing organism that can withstand whole body freezing. So it freezes into essentially an ice cube. It has no heartbeat, no brain waves. It's essentially, for all intents and purposes, dead. It's in suspended animation through our cold North American winters. And then come the spring, it miraculously thaws and hops to a pond where it mates. So I heard him giving this talk and I was just like, wow, this is amazing. How does this frog survive this and go and hop into a pond that's chock-a-block full of microbes and stay healthy? So I really wanted to go and work with him to understand the wood frog and how its immune system is modulated despite undergoing freezing and huge suppression of its metabolic processes. So that's kind of where I ended up. So I moved to Ottawa and did that postdoc with Ken. And then while I was there, as a postdoc, you always need to be looking at your next step ahead. Mm-hmm. So I applied for the Banting Postdoctoral Fellowship at the University of Waterloo. At the same time, I had applied for the assistant professor position at the University of Waterloo. And both the responses came back within a few days of each other that I had been awarded both.
Oh, wow.
So that was really exciting, but it meant, what do I do? Now I have a choice. Right. And it's a good problem to have, of course, but sometimes decisions are hard. So what I actually ended up doing was deferring the start date of my assistant professor position by about a little over a year so that I could hold the Banting Fellowship. And during that time, I collaborated with some other researchers at the University of Waterloo and began establishing some of the research projects that I carried forward into my faculty position that I'm working on now.
That is phenomenal, Barb. Thanks so much for talking us through how you got to where you are today. And we mentioned in the beginning of our interview that you are studying a lot of different topics, many of which are just absolutely amazing. So is there one that you are the most excited about right now that you want to share with us in more detail?
That's tough because I do have a lot of excitement about all of my research projects, and that's kind of why I choose them and pursue them. But I suppose if I had to pick just one, it would be the research that we're doing right now on developing a system to study the role of the skin epithelial cells on frogs in acting as an immune barrier. So understanding how these skin epithelial cells are able to sense the microbes in their environment and relay that information to the underlying immune cells in the skin. So the skin is really important because it acts as a physical barrier. So it keeps everything in the outside environment out and everything on the inside. But it also plays a really important role in chemical defenses. So I mentioned those antimicrobial peptides earlier, and these are really short molecules that are synthesized by glands in the skin and then secreted out onto the frog's skin. And for a long time, we've regarded them as chemical weapons that the frog secretes to inactivate pathogens that are on its skin. But we're starting to understand now that they might actually have a role in acting on those very same host epithelial cells and telling the host epithelial cells how they should respond to the pathogen. So that's something really exciting that I'm looking forward to getting more answers from.
Interesting. Now, is this in that North American wood frog as well?
Yes, they actually do have antimicrobial peptides. So far, we think there's probably only 1 or 2 families that are expressed on their skin or secreted onto their skin, but we're also searching to see if there are any others that we've perhaps missed.
Very cool. So you're getting closer to unveiling the mystery of the North American wood frog and how it does this amazing transformation.
We are trying.
I love it, Barb. Well, this is a really cool project, and we've talked about a lot of the great aspects of your career. But as you mentioned, when we were talking about motivation, I think failure is also really common in science, and it can be motivating, but it can also be such a struggle and such a downer, I think, on some days when it just feels like everything's going wrong. So do you have a moment of struggle or challenge or failure that you can tell us about today, and maybe talk us through how you worked through it, since some of our listeners may be in the same boat with you?
I would say the moment that still sticks with me even to this day was during early days in grad school, where I was trying to do this one experimental technique. I needed it, or I thought I really needed it for my project to keep progressing. And I was trying it 6 ways from Sunday for months. And I wasn't making any progress. So I was facing constant daily failure.
Right.
And I got into this spiral of, I thought if I just tried harder, so staying late in the evenings and the weekends, and just kept trying different variations, that I would get there, right? Perseverance. But after months of doing this, I don't know, there was just one day that I'd had enough. I came in, I remember it was a Friday morning, and I was waiting for my supervisor to show up so that I could walk into his office and tell him that that was it. I was going to leave grad school.
Oh, wow.
And at the time, my supervisor had a secondment at the School of Public Health. So he would spend his mornings at his office over there across campus. And then he would come into the afternoons to the Department of Biology and spend his afternoons there. And for whatever reason, he just didn't show up that day. So I couldn't go into his office and quit.
He knew, right?
I don't know if he knew. I'm sure he was just busy with something, but he didn't show up. So I couldn't go in and quit.
And then you had the weekend to recover, right? By Monday, you were better.
I didn't go into the lab, and I chatted with some of my really good friends and let them know how I was feeling. And taking that time away really gave me some perspective. It made me realize that it was important to take a step back and actually see the forest and not the trees. And that, did I really need this one component, or was there some other way that I could work around it and get to the end without having to go through this problem? So giving yourself some space and time to actually reflect on what you're doing and to reach out to others when you're struggling, because everyone's in the same boat. It's hard for everyone. Some people are just better at hiding it than others. So don't let yourself get hung up on a problem. Just keep going around it. You'll get there eventually.
Definitely. So did you ever go back and tell your advisor about this crisis moment that you almost had?
I never did, actually.
Really?
So hopefully he doesn't listen to podcasts and hear this. But it all worked out in the end. And I'm really glad that I didn't quit, because I really do love science. I love what I'm doing. And had I let my feelings in the moment get the best of me, I might not be here in this position, but I'm sure that my supervisor would have chatted with me and he would have come up with a plan. And I don't think he would have let me quit so easily.
So Barb, did you ever figure out why it wasn't working, or did you ever get it to work?
I didn't.
It's one of those unsatisfying, lingering things in your memory, right?
Maybe that's why it sticks with me for so long. But I just said that instead of doing that, I did something different. And I molded my project in a slightly different direction, and it worked out. I think sometimes maybe we just don't have the tools to do something in that moment in time. And you just kind of have to do what you can and move forward.
Definitely. And I think sort of pulling the plug on a project that you're that invested in and maybe that excited about is one of the hardest things in science, but it's something you're absolutely right that you do have to do sometimes because it can absolutely stall your progress and really keep you from meeting your goals when there are other ways to get around it and other ways to get there. So such an important message for our listeners. Thank you for sharing your story. But we don't just want to talk about your difficult times, Barb. Can you tell us next about one of your exciting successes, whether this was a huge win or even just a small one that really meant a lot to you personally?
Probably one of the most recent ones is that we've submitted a manuscript with 2 of my graduate students as authors on the paper. So for me, that's a big win because I really enjoy seeing my students grow and contribute to science. And it was a big win for them. So it kind of ends up being a big win for me because I live vicariously through them now, right? Another one would probably be one of my master's students who's just started, actually went to a conference to present his initial findings, and he won a best poster competition prize, which was so exciting for both of us. So they're all doing phenomenal. My wins are, I guess, my students' wins, really.
I love it. Well, I think you're absolutely right because your students are the people that you're training. Watching them grow up and have these exciting accomplishments and milestones of their own career can be so rewarding. So I have to ask, how do you celebrate in your lab when your students get one of these big wins?
I suppose we maybe follow in the tradition of when I was a PhD student with Mike Belosevic, in that we normally go to the on-campus watering hole and have a celebratory beverage.
Awesome. Well, I think it is so important to take a moment to celebrate and to acknowledge the hard work of your students and all of the great things that they're doing in the lab. And we've talked about a variety of different aspects of your career, but one of the things we haven't really gone into detail on are some of the opportunities that you've had to travel. You mentioned this recent conference where your student won a Best Poster Award. And I think these conferences can be great opportunities to present your work, to connect with people, to meet some of these scientists that you've just been reading about in the literature. So do you have a favorite place, Barb, that your science has taken you?
There's lots of favorites, but probably where I had the best time was in Prague in the Czech Republic. We went there for the 2009 International Society for Developmental and Comparative Immunology conference. And it was a group of us from the University of Alberta that went. And I just thought, sitting there in the audience, this was my first real big international conference in my area. And I was sitting there listening to the presenters. Many of them were people whose papers I had read, and I was so in awe and so inspired by the science and getting to meet these individuals whose papers I had only ever just read in person. So that was fantastic. The science was great. Networking was great. And then the group of us also took a few days before and after the conference to go traveling around different places. So we spent a couple days in London beforehand. And then one of my best friends from grad school, who's still my really great friend now, we went traveling to Berlin and Netherlands afterwards. So it was really a good combination of science, but also some personal fun.
That is fantastic. And I love that you mentioned this almost starstruck feeling when you got that moment like, oh my gosh, it's Smith et al.
I know. Our people that we fawn over, our science celebrities, like, oh my goodness, should I go and talk to them? Would they want to talk to me? And of course they do. They're all about the science. So they love to talk to anyone who wants to talk science.
Absolutely. And you mentioned, I love that you were able to tack on some extra time to travel while you were overseas for this trip. So did you have some favorite places that you visited, either in London, Berlin, or Netherlands?
For me, Berlin was really eye-opening because it was a mix of these new buildings, but also really old structures. And in Berlin, they've left a number of buildings that have damage from the world wars to them still there in place. So as you're walking by, you still see all of this. And it's always a constant reminder to everyone of the past and what has happened. And they had just some wonderful exhibits that were really well done to highlight the Holocaust that happened there and just some of the horrific things that happened as a result of the genocide. So it's really good to go there and see it in person instead of just reading about it in books.
Definitely. And I think that sense of history in a lot of these European cities, it really leaves a lasting impact on you when you get to visit. So very cool to hear about some of your travels for science. And we've talked throughout our conversation about some of the wonderful people, Barb, that you're able to work with as a scientist. And I know there are stereotypes out there about what scientists are like and what life as a scientist is like. But I love to try and break those on our show by talking about some of the fun and funny things that scientists do when they're together, just to show that there is this fun, creative, amazing side of life as a scientist. So do you have a story of your own that you can share that really highlights this human side of science?
Oh my goodness. These are maybe some embarrassing ones. One that happened to me personally would be during my PhD, we had an aquatic facility and we had these very large tanks that we would hold our goldfish in, and they were up on a platform. So as a little personal thing, I find clothing and I love it. So then I'll wear it all the time to the point that it nearly wears out. So I'm sure you can see where this is going. But these large, giant tanks of goldfish sit up on a platform that you would have to take a large step up onto. So one day I was wearing my favorite pair of jeans that I wore almost every day that I had really conditioned, really worn in. I took this giant step up and all of a sudden there was a
No. Even the goldfish are cringing, right?
Even the goldfish are cringing. They're averting their eyes. They're like, oh no, this is horrible. Luckily for me, it was also a requirement that while you were in there that you had to wear a lab coat, which hid the rip until I was able to run back upstairs to the lab where I conveniently had a pair of running pants because I was going to go for a run later that I could change into. Crisis slightly averted, but embarrassing.
Listeners, this is just pointing out again the virtues of exercise is always having backup clothes on you. So.
Exactly. I was so glad I was a runner that day.
So was anyone else in the lab to witness this?
So they weren't in the aquatic facility with me. But as I came upstairs, yes, the lab room was filled with other grad students. And as I was reaching under my desk, they were like, what are you doing?
Nothing to see here.
Nothing to see here. And I had to sheepishly admit that I had ripped my pants.
Well, I am sorry about the loss of your cherished jeans there. I must say that is always a tragic event.
Yes, they went straight into the garbage bin after that. There was no attempt at fixing them.
Well, I think this really does highlight just the fact that scientists are normal people. These weird, funny, and sad happenings are very common for scientists as well. So we're not these little robots just working in the lab.
No, we have all the same unfortunate events happen to us.
The comedies and tragedies.
That's right.
Well, Barb, we've talked about a lot of the great work that you're doing as a scientist, but I know there's always things standing in the way between you and the questions you would love to answer, whether that's funding or not having enough staff or the technology's not quite there, or maybe just the project isn't feasible. So if I gave you everything and took away all of the barriers, is there one question, Barb, that you are just the most passionate about and would want to answer first?
I try not to let those things be barriers for me. So I would say probably that this amphibian skin project is the thing that I'm most excited about at the moment. And so I try to push that forward despite challenges. But another facet of science that I would love to go back and revisit was some of the initial studies that I started doing during my postdoc at Carleton University, where I was interested in examining how different processes that affect the wood frog, that one that can freeze, so like freezing and then the process of thawing, how that affects the cells in the bone marrow of the wood frogs. And the bone marrow is really important for frogs because it acts as the hematopoietic niche. And that's just a fancy phrase for saying where all the blood cells, so where the red blood cells and your white blood cells that are important for your immune system are generated from stem or progenitor cells. And I really wanted to look at that to see what was happening there, both the cellular processes and the molecular components that were being changed in response to freezing and thawing. So I'd love to go back and revisit that once I have more money and more people to do it.
Well, this is a fascinating project. And I love that you brought up the amphibian skin project as one that you're actually so excited about and also able to work on. So I want to challenge you a little bit here. If we gave you more resources, more time, more students, more whatever you need, how would you enhance that project?
Well, right now we're trying to make cell lines from amphibian skin, and we've been somewhat successful, but I'd love to expand our repertoire of cell lines. So right now we've been focusing on just a couple of frog species, so the wood frog, but also the African clawed frog. But there's nearly 8,000 different frog species out there. So studying just a few doesn't necessarily give you a picture of the diversity that exists within the frog species as a whole. So I'd love to be able to expand it. I'd love to be able to diversify the types of pathogens that we're looking at as well. So right now we're studying frog virus 3, which is one of the diseases that are causing mass mortalities in frogs, both tadpoles and adults. And I'd love to be able to expand just the different types of viruses, bacteria, fungi that infect these amphibians and study how their cells respond to that. And I'd love to be able to see exactly what's happening inside these cells by doing things like RNA sequencing at different cells, different pathogens, different time points, under different environmental conditions. So at temperatures that the frogs normally exist in, but then also temperatures that are much higher than what they normally live in as a proxy for climate change, but also temperatures that are much lower because climate change is giving us these rapid and extreme shifts in temperatures that amphibians will experience. And amphibians are different because they are ectotherms, which means that their body temperature is that of their environment. So whatever temperature their environment is, that's what their body temperature is. So a lot of their cellular processes, such as their immune system, can be heavily influenced by the temperature that they're living in.
Certainly. Well, Barb, it sounds like there are so many cool questions left to be answered on this topic. So you've got your work cut out for you, I think.
I think I do, but good thing I have the next 30 or 35 years to do it.
And all of the students to help you along the way.
That's right.
Well, Barb, you've given us so much insight and information and things to think about throughout our conversation today. But there's one more thing I'd love to ask you for, and that is advice. Can you share with me and our listeners a piece of advice that you were given that really had a big impact on you and your career?
I would say to focus on the now instead of worrying about the future, because things do have a way of sort of falling into place. And that definitely helps with those anxious feelings of, well, I don't know what's going to happen in the future. And you can't control that. So just focusing on what you can work away at now often has a calming effect, which I think is something that we in science all need. And probably a lot of people that aren't in science, when they're facing uncertainty and they don't know what their future is going to look like, I would say focus on the now.
Wonderful advice, listeners. Definitely take this to heart. Don't let that anxiety take over. Yeah. And Barb, can you share with us, is there any other last piece of advice you'd like to give us, or a last final message you want to leave our listeners with today?
Well, I think I've already talked about the idea about failure being okay, and it's part of the learning and growing process. So I think maybe the last piece of advice is do what you love if you can. I believe we're all inherently good at something. We have an inherent ability, and it's about searching out and finding what it is that you do best and then pursuing that.
Excellent advice for our listeners, Barb. If they want to learn more about you and all the wonderful work that you're doing in science, where should they go, or how should they get in touch with you?
I would say the best way to learn more about what we're doing and also information on how to contact me is through our lab website. So if you were to Google Katzenback Lab, probably be the first thing that pops up. And I know Katzenback is a little bit of a odd last name, so it's just spelled Katzenback.
I think it's a lovely last name, Barb. Well, listeners, definitely check out Barb's lab website. She's got some great information on there. And Barb, thank you so much for joining us today and sharing a piece of your story.
Oh, thank you so much, Marie, for having me. I really enjoyed chatting with you.
Well, it was so much fun to chat with you as well. And listeners, as always, great to have you with us. We'll see you next time on another episode of People Behind the Science.